Good morning, everyone. I'm Christopher Clemmensen, the group leader at Center for Basic Metabolic Research, and it's my great pleasure and honor to introduce the next speaker, Professor Laurie Seltzer from Columbia University. So, Laurie graduated from Princeton University, did a PhD at Rockefeller University, did postdoctoral work at King's College London, and returned to Columbia University, and she's done tremendous work for many years on periphery brain crosstalk mechanisms pertaining to energy homeostasis, feeding behavior. And recently, Laurie was also promoted to full professor, so there's a little bit of an update to the introduction, and congratulations. And today we're going to hear about some interesting work, and I look forward to hearing about sympathetic nervous system regulation of brown adipose tissue function. So please, Laurie, the stage is yours.
So I'd like to thank the organizers for inviting me today. It's really a pleasure. So my lab generally studies neural circuits that regulate appetite and metabolism. For the large part, my lab has really been focusing on central circuits. But today I'm going to talk about a completely new area of research for my lab. We're still developing a lot of the tools, which focuses on the periphery, so nothing is published that I'm talking about today. And what I want to talk to you, because this is a pretty diverse audience, which I'm super excited about, I want to talk to you a little bit about why it's both interesting and clinically relevant, potentially, to study sympathetic regulation of metabolism, focusing on brown adipose tissue, or BAT. And I'm going to also talk to you about the trials and tribulations and progress that we've made in developing the tools and assays that are actually needed to study this appropriately. So I don't need to introduce this slide in this room, but just to say that this is a starting point, and what intrigued me about the brown adipose tissue system was studies that were done using PET imaging with a radio-labeled glucose tracer injected into patients, where it was noticed that cold stimulated brown adipose tissue, glucose uptake in brown adipose tissue in lean individuals, and this cold stimulated glucose uptake was decreased in people with elevated BMI and obesity. And so this type of observation really set off a whole cottage industry of people who were looking to use BAT activators as therapeutic anti-obesity targets. And both the clinical data and the basic research data sort of showed over time that this relationship is much more complex. And I think really the best way of highlighting this is a recent study that was published a couple years ago now by Paul Cohen at Rockefeller. And what he did is they did a huge retrospective study of over 50,000 PET scans for cancer patients. And so these patients were not cold exposed. This is just baseline brown adipose tissue activity. And what he did was he basically stratified all of the patients into BAT positive or BAT negative patients, and then analyzed a whole bunch of different metabolic profiles. And what he found is, so the patients that had the presence of active BAT, shown here in brown, so they had lower glucose at baseline. Now, this effect on glycemia was really pretty modest. But when he stratified by BMI, now you can see an interaction between the BAT and the elevated BMI, where at low BMIs, you don't really see any effect of the brown adipose tissue activity, but at the high BMIs, now you start to see a protective effect where those individuals had lower glycemia, and this also tracked through to prevalence of type 2 diabetes. So also the individuals, you could see some modest protection of glucose intolerance in the patients that had brown adipose tissue activity, but you really start to see a large effect size in the context of a BMIs over 30. And so this raises the possibility that maybe clearly brown adipose tissue activators are not sufficient to prevent obesity. As you could see, there are plenty of people who had BMIs over 30 that in fact also had active BAT, but it may be really useful as a target to improve metabolism in the context of obesity. And so I showed you before the PET type of approach to study glucose uptake as a signal of active BAT. Another way that people have studied activated BAT in humans is by direct thermography, because when BAT is activated, it also generates heat, and so this has also been used. But while these studies have been really interesting and intriguing in humans, in the end, if you really want to understand what's going on and what is the relationship between obesity and metabolic dysfunction and brown adipose tissue, you really need to get to a more mechanistic model, which brings us to rodents. And so this is basically a summary of what is known about the circuits regulating thermal regulation and brown adipose tissue thermogenesis in rodents. So these are pretty well mapped out by many different labs. And so what happens is thermal sensory information, so both heat and warm information, is relayed and processed in the hypothalamus. These signals then get relayed into the hindbrain and out through the intermediate lateral column of the spinal cord and into the sympathetic ganglion, which finally you have the sympathetic neurons that then project out to brown adipose tissue. Now, the vast majority of research that's been done in thermoregulatory circuits has either focused on the circuits in the brain, which have been now the populations, in some populations regulating thermal control are really quite nicely defined, or they've focused out in the brown adipose tissue depot itself. And what my group is focusing on is rather the projection from the sympathetic ganglion out to brown adipose tissue. And so there have been, because of this link between BMI, elevated BMI, diabetes, impaired brown adipose tissue functioning, so there have been many groups that have used animal models to try to understand what is the link between elevated BMI and impaired brown adipose tissue function. And basically, depending on where you look, there have been many different cell types and mechanisms implicated. And so the brown adipose tissue is a complex tissue. It has brown adipocytes. It has various immune populations, including macrophages. Their cell autonomous mechanisms have been identified in both adipocytes and in the macrophages that are thought to confer resistance to norepinephrine, the main signal of the sympathetic nervous system. There's also evidence that both the innervating fibers as sympathetic nervous system fibers, as well as the vasculature, also remodel in response to obesity, and that the density of the fibers and the vascularization is decreased. There's also evidence that sympathetic tone coming from the brain is also decreased. And some of these, it's possible that some of these mechanisms may be acting in concert or in different circumstances, although some of the mechanisms are also mutually exclusive. And part of the reason why we really can't understand what the relationship is stems from the approaches that are currently available to interrogate this issue in rodents. And so first, one classical example is when either the nerve innervating, coming in, innervating the brown adipose tissue, I don't think this works, but the ones that are coming in at the side, so those nerves, either you can denervate those nerves or you can stimulate those nerves. A lot of people have done that. And when you do that, the problem is that both sympathetic and sensory nerves are innervating the brown adipose tissue through that mixed nerve population. And because those two cell types are thought to have opposing effects, if you're either denervating both or stimulating both, it's obviously complicated to interpret. So other types of approaches that have been used to study this issue, a classic one is to use pharmacological stimulators, so either to inject peripheral injections of either norepinephrine or various beta adrenergic receptors. And what you can see, an example on the left, is if you inject either norepinephrine or the beta-3 adrenergic receptor, you nicely induce bat thermogenesis, measured with a temperature transponder. You can also, on the right side, inject a beta agonist and also induce glucose uptake, specifically in brown adipose tissue and not in liver. And so these are the types of assays that are very typically used in rodents. But the problem is if you actually want to understand where this resistance to the sympathetic tone is coming from, if you just come in with the agonist, you've now ignored all of the central circuits, all of even the peripheral circuits, and you go in, you directly activate the adipocytes and the endothelial cells and other cells within the depot. And so you're really sort of shortcutting any regulation that can happen, so it's hard to understand what's happening endogenously. So another approach that's often taken is, so instead of jumping right in and attacking the adipocytes directly, so now you expose animals to cold for a while. And here's an example of mice put in 4 degrees into a cold challenge, and now we're measuring brown adipose tissue heat production directly with thermal imaging. And you can see again, if you move mice into the cold, you can get this nice induction of bat thermogenesis. But the problem is, again, when you are moving mice into the cold, you're inducing a systemic response. So yes, you're inducing changes to brown adipose tissue, but you're inducing changes to white adipose tissue, to the general circulation. And again, you can't really study and isolate what's happening at the level of brown adipose tissue. And finally, the last approach that people use is to look at tyrosine hydroxylase. So tyrosine hydroxylase is the rate-limiting enzyme for the production of norepinephrine, again, the major neurotransmitter of the sympathetic nervous system. And what people do is they either do like a Western blot in bat for tyrosine hydroxylase expression, or they could also do what's shown here is IDISCO immunolabeling in an intact depot in conjunction with whole organ imaging to get a reconstruction of what the fibers look like. And so you can see here really nicely the big nerves that are coming in out on the lateral edges of the depot and then sort of branching inwards. So people characterize that density. But again, if you're looking at just a neuroanatomical organization, or even more so, you're looking at a single point at a Western blot of tyrosine hydroxylase expression, it's just not clear exactly how that correlates with activity. And today, neuroscientists have much more precise tools to measure these activities in real time. And so when I set out to study this problem, I decided that I wanted to build a toolkit to study sympathetic regulation of brown adipose tissue activity. And the key is that it's easy to use. Because right now, there are some labs out there that have very specialized techniques, but those techniques are either not broadly available or are too expensive, and so there's a high barrier to entry. And so I really wanted to develop tools that could be readily adopted anywhere so that we could all start studying the same things and start comparing notes. So the key items I wanted to develop is one, an assay to really precisely measure bat function in vivo in real time, and the other was to get genetic access to specifically modulate neuronal innervation of brown adipose tissue. And so with the first developing the in vivo assay, I decided to start with an anesthetized prep, just so there would be more control over the temperature and more consistent outcomes. But I was well aware, and I'm sure, again, maybe many people know from the work of Barbara Cannon and Jan Nedergaard, that isoflurane, which is the anesthetic most commonly used for these types of surgeries in animals, suppresses brown adipose tissue activity. So I compared various anesthetics, and all of this is just direct temperature recordings from brown adipose tissue in response to saline in blue versus the various anesthetic. And you can see with isoflurane that brown adipose temperature is reduced. In contrast, in ketamine, xylazine, which is Columbia University's second-line anesthetic of choice, so I had to show them what would happen when I use this, and you can see conversely that we get increased brown adipose tissue thermogenesis. And what we did find after screening a number of anesthetics, that if we deliver Avertin through constant infusion through the tail vein IV, that we're able to maintain a very consistent level of plane of anesthesia for about 2 hours. This consistency is also important, is because if you have mice waking up and then you're putting them back down and waking up, of course, brown adipose tissue temperature and body temperature is going to fluctuate. So really maintaining a stable plane of anesthesia is critical. And so just to introduce you briefly to the sort of setup that we use, we have mice that are on a platform, again, anesthetized with Avertin. We have a probe, a rectal probe, measuring core body temperature. Then we have another probe measuring lower back body temperature, and a third probe measuring BAT temperature. And what we do, the calculation that I'll show very often, is this delta BAT minus lower back, and that is what we call brown adipose tissue thermogenesis. And so this is just to show you what happens in this system when we add in norepinephrine. So you can see in the first box, looking down in the baseline condition, nothing happens. Then we inject pre-warmed saline. Again, nothing happens to the recording. When we inject norepinephrine, you see this big spike, and you see an increase in actually both the core body temperature, the lower back, and the brown adipose tissue. This is just to remind you again that when you're adding in norepinephrine, you're affecting the entire body. But when we then follow up with a beta blocker, propranolol, then we're able to completely block this, the thermogenesis. So that's great. We can measure this with norepinephrine, but I already argued before that norepinephrine is really a sledgehammer and acts through everywhere. And so we wanted to make sure that this system is actually sensitive enough to detect endogenous and physiological changes in body temperature. And so now what we did is remember the mice are kept on this platform. The platform is warmed to 35 degrees. And we get a baseline recording. Then we just shut off the heat and let the temperature naturally fall down so that the platform temperature hits about 31. Then we do another series of recordings, and then we turn the platform back on. And so what happens when we do this is, and you look at core body temperature, not surprising, when you turn the heat off, the core body temperature falls, and when you turn the heat on, then the core body temperature comes back. The lower back temperature exactly follows the same pattern as core body temperature, but the brown adipose tissue does not. It maintains a constant temperature. So when you look at this delta BAT minus lower back, you can see that we turn just the natural cooling of the body is associated with increased BAT thermogenesis, and this turns off when the heat on the platform is turned on. And what you can see is this doesn't happen immediately with the heat, with the change in the platform, but rather it requires a drop of about 0.8 degrees before we see brown adipose tissue coming on. And then on the flip side, when we turn the heat on, it seems to be more sensitive, even within less than a half a degree of an increase in core body temperature, we already see brown adipose tissue thermogenesis turning off. And so this system really reflects the very tight body temperature regulation that normally exists in all organisms. So we felt confident that we had this system. With this in play, we not only looked at thermogenesis, but we also looked at blood flow with the Doppler imager. And the blood flow follows exactly the same pattern now in the blue as we saw with the thermogenesis, is when you inject norepinephrine, you get this big spike in blood flow, and that is completely blocked, or mostly blocked, I should say, using the beta blocker. So we were happy with this assay. We have something to start with. We're also working on in vivo systems to measure norepinephrine and also nutrient uptake. But then we decided to move with this assay in hand over to get genetic access to specifically modulate innervation of BAT. And so this is just a diagram of the sympathetic nervous system. So the sympathetic nervous system, all of the ganglia basically lie parallel and adjacent to the spinal cord. And the sympathetic ganglia, the neurons, project out to the various organs, and it's really organized very much in a sort of rostrocaudal map. And so we want to just target not all of the sympathetic projections. We want to only target those projections that go to brown adipose tissue. And so this is just showing you in vivo data from a postdoc in my lab when she was back, Maria Francois, when she was back in Heike Munzberg's lab. But what you can see in the TH stain, so that this is a TH reporter line, so this is actually a sympathetic chain, again, lining the whole spinal cord. And if you inject a GFP-labeled pseudorabies virus, you're able to retrogradely, that goes from the brown adipose tissue into the ganglion. And you can see that only a few of the ganglia are labeled. And in fact, quantifying it out, more than 50% of the sympathetic innervation of brown adipose tissue comes from the stellate ganglion. So now we use two different strategies to try to isolate and activate only those projections to brown adipose tissue. So we used an optogenetic approach and a chemogenetic approach. And so because this is a diverse audience, I'm just going to give a brief overview about what optogenetics and chemogenetics are. And so basically, they're both very similar conceptually, which is if you focus on the middle, what you do in both cases is you're able to achieve targeted expression of a receptor that doesn't have any endogenous ligand in the mouse. And then what you're able to do is you then activate, in the case of chemogenetics with the ligand, in the case of optogenetics with light, and you're able to activate just that circuit. So in the case of chemogenetics, there's a genetically altered muscarinic receptor, also called the DREAD receptor. And what you're able to do, it's coupled to a GQ, in this case for stimulation. And if you just express this DREAD receptor on its own, absolutely nothing happens because there are no endogenous ligands, again, in the mouse. But if you inject with CNO, then you're able to stimulate just those neurons that express the DREAD receptor. And optogenetics is basically the same idea, except instead of expressing a receptor, you actually express a channel rhodopsin that senses blue light. And so in the absence of blue light, this is completely inert. But if you shine blue light, you're now able to activate just those neurons. And so we started with an optogenetic approach because what you're able to do is to just take a TH-Cre mouse line and drive Cre-dependent expression of the channel rhodopsin. And so now you would be driving channel rhodopsin expression in all of the sympathetic nervous system. And in order to target just the fibers going to brown adipose tissue, so we worked with the Sun Lab to develop these sort of these small blue light adapters that would shine light just on these nerves as they're entering brown adipose tissue and activate them. And so Sung-Il Park, again from Texas A&M, is the one who developed this system, and he had already used this successfully with Carlos Campos to activate peripheral projections in the gut. And so we did the same thing in brown adipose tissue, but when we used the optogenetics and we turned on the blue light, basically we saw absolutely nothing. And so if anyone's interested, I can speculate about why that happened, but we decided after working on trying to optimize it a fair amount, we had moved on to chemogenetics, which is a little bit trickier. So with chemogenetics, again, we want to express this dread receptor, and now, but we needed to express it. The advantage of the optogenetics is you could just target the light on BAT. So here with chemogenetics, we had another problem. We want to just target the sympathetic neurons that are innervating BAT. So we wanted to use a retrograde tracing approach. So the first thing we did is, so I had showed you before, let's see if I can go backwards, that when Marie injected the PRV labeled GFP into brown adipose tissue, that you can very specifically retrogradely trace to some parts of the sympathetic chain and not all of it. So that's really key because we want to make sure that if we're tracing back, we're not hitting projections to the white adipose tissue, to the pancreas, all of these other metabolically relevant organs. So that's really important. But the next thing that we needed to make sure of is the stellate ganglion, which is the ganglion that sends the majority of the innervation to brown adipose tissue. We needed to make sure that the stellate itself projects to a lot of different organs. It projects to brown adipose tissue, to forelimb, to heart, to lung. We needed to make sure that if we're targeting a neuron with a retrograde injection from BAT, that we're only hitting the projections to BAT and not to the heart or to the forelimb. And so what we did, I'm just going to show you one example here. We injected another retrograde trace, chlorotoxin B, conjugated to a fluorophore, so we conjugated it. The red fluorophore we injected into brown adipose tissue. The green fluorophore we injected into the forelimb. And you can see that the neurons innervating brown adipose tissue and forelimb are completely non-overlapping. They're also non-overlapping from the neurons that project to the heart. And so that now with this information, we're pretty confident that at least with the retrograde approach, we would be able to inject a retrograde virus into the brown adipose tissue and that it would go into and only affect those neurons, sympathetic neurons that are projecting to BAT. So that was the plan. But when we injected and we tried a number of the standard retrograde viruses that are available, various serotypes, and including most of the ones that we use in the brain without a problem, what I want you to see, the comparison here is the injection of the chlorotoxin B versus the retrograde trace. And the retrograde trace with the existing serotypes were just not good. And we just could not get enough infection in order to really get decent expression and begin functional assays. So we worked with Viviana Gradunaru's lab at Caltech, who developed a series of new serotypes that work better in the peripheral nervous system. And we tested her serotype, and in fact, you can see on the right that now we get really nice, robust expression in the stellate ganglion in the sympathetic nervous system. So this is great, but there's now another issue that cropped up, which is that I told you way back in that diagram of brown adipose tissue that brown adipose tissue is innervated by the sympathetic nervous system and the sensory nervous system, and it's thought that they have opposing functions. And so we needed to make sure we were not hitting when we were doing our retrograde injection, if you just inject the retrograde virus here conjugated to GFP, you can see that not only are you labeling the stellate ganglion, but you're also labeling the sensory neurons. You're labeling the neurons in the GRGs at the C5 and T1 level. And so this type of stimulation is not going to work because, again, you're going to hit the opposing populations. So we next added in a Cre-dependent step. So now we had the Th-Cre neurons, the driver that it's expressed in the sympathetic nervous system. We added a Cre-dependent retrograde virus with the Gradu-Naru serotype. And now you can see that we're able to get really nice expression in the stellate and not in the sensory ganglion. And just to say that the cellular distribution is a little different here because this is actually the expression of the DREAD, and the DREAD is a G-coupled protein receptor, so it's actually localized to the cell membrane. Okay, so finally we get to do our experiment. We now have the expression where we want. We inject CNO, and now I'm just showing you core body temperature, blood flow, and bat thermogenesis. And so we inject again, the baseline in salines are really nice and flat. We inject CNO. We're able to see a robust and increase in both bat thermogenesis and blood flow that is blocked by propranolol, the beta blocker. And this is just to show you, if you take a virus that expresses just the GFP and not the DREAD, and you inject CNO and propranolol, you have absolutely no effect. So this is very specific now for just the sympathetic innervation of brown adipose tissue. We did the same experiment with an alpha blocker. So alpha blockers, the bat thermogenesis is thought to be regulated primarily by beta adrenergic signaling. So when we add in the alpha blocker now, phentolamine, you see that the effect on thermogenesis is really not blunted. But if you look at blood flow, now you can see that the blood flow is decreased by about 50%. And so again, just comparing things, this fits with what has already been known in the literature about regulation of blood flow, which has alpha and beta adrenergic components, while the bat thermogenesis is only beta. So all of this has been in this anesthetized prep. We're now in the process of moving to in vivo in awake animals to see whether we can also impact systemic homeostasis. And so this is an example of a mouse now that has, where we inject the CNO into mice that now, in conjunction with an oral GTT, and you can see that chemogenetic activation of only sympathetic innervation of brown adipose tissue is able to improve glucose tolerance. And we've also, the data is a little bit too fresh to show, but we've done the same thing now in the context of indirect calorimetry, and we show that we can get increases in energy expenditure and also changes in RQ, also induced by this type of thing. And so we are again feel good about the fact that we now have a system where we can get access to chemogenetically stimulate both brown adipose tissue thermogenesis and blood flow. And now we're again starting to work on more of the endpoints in awake mice. And in the long term, now that we have all of this sort of almost nailed down, we really want to understand how the question I posed in the very beginning, which is how are responses to sympathetic signals altered in the context of obesity, diabetes, and aging. With that, I will thank my lab. Daniele, who was Daniele Neri, the PhD student who really spearheaded all of this work, and funding organizations as well. And as well as my collaborators from both Caltech and Texas A&M. I'll take any questions. Thanks.
Thank you, Laurie. Fantastic presentation on trying to take some of these well-known tools from the brain biology field and try and apply them to study bat biology. Very interesting. I'm sure we have questions from the audience over here.
Yes, hi, Haley Berm. Really nice talk. I was just curious with the chemogenetics, did you ever or do you plan to do any continuous dosing of CNO to see if there's any like desensitization or if you can, keep that activation going for longer periods or even with like DCZ and like
Right. So we're working with DCZ right now because DCZ all of our initial studies we had done with CNO. DCZ works at about at least 100-fold lower concentrations, and it's easier to do those types of dosing experiments. And so we're going to look into using DCZ in the context of like a mini pump or something where you get more continuous low-level infusion, and we're going to see if we get that type of tachyphylaxis or not. But that's in the offing as well.
More questions? This in the front, if you don't mind passing down the microphone.
Fantastic presentation. I really got some ideas. And do you intend to somehow evaluate the mitochondrial function of this brown adipose tissue, like in real time, because it's thermogenic and so on?
So we are starting to work. So there are a number of tools that are coming online. We weren't originally. I mean, the mitochondrial function wasn't on the first thing, but it will be one of them. But one of the things I had sort of alluded to in just the summary slides, we want to look at actually norepinephrine release is actually one of the very top things that we want to look at. There are various GRAB fluorescence sensors that have been used in the brain in order to measure and visualize norepinephrine release. And the problem, which we're working with some collaborators to overcome in the periphery, is that in the brain they have these, they mount these little mini scopes or fiber optics right onto the skull, and this has the real advantage that the fiber optics moves with the animal because it's mounted to the skull in brown adipose tissue, especially because it's like right over the heart and in the part of the body where the animal is, and the lungs where the animal is breathing. So doing those measurements where you have all of that movement is the issue that we're trying to overcome right now, and hopefully we'll be able to get there. But if that can be overcome technically, then it opens up really the door to a lot of things, not only things like norepinephrine sensors, but also some of the fluorescent mitochondrial sensors, ROS sensors. I mean, there are a ton of tools out there that have genetically encoded sensors that we could use, but we need to overcome that obstacle of the vibration.
More questions from the audience? Maybe meanwhile, I'll briefly ask if have you given up on the optogenetic approach? You mentioned a little bit that you had some reasons for why you thought that didn't pan out, but maybe you can elaborate.
Well, we had played around with a lot of different stimulation protocols and intensities, and for one thing, if you turned on the intensity too high, you just heated up the brown adipose tissue on its own. So that really didn't work. Brown adipose tissue also expresses some opsins, so you need to keep the stimulation pretty low. It's possible now, looking back on it, that there are reports that tyrosine hydroxylase is transiently expressed during development in some of the sensory neurons. And it may be because we generated those mice by a cross, so it would be a developmental expression. And so it's possible. At the time, we were really focusing on just validating and looking at the stellate to make sure it was expressed. We weren't really looking back at the DRGs back then, but it's possible that what we had is an issue of expression with the sensory nerves as well that are acting in opposition. And so we have, we're in the process of validating another line that is TH, that is not TH, that would be able to target just the sympathetic innervation. And so we're hoping that it may be, if that is validated, we may revisit the optogenetics, because going back to the issue that was asked before about getting continuous stimulation, it's really, if you have an optogenetic, they have LEDs in there that you can implant, and you can program the LEDs to stimulate with different time courses. And that would be much nicer than just using a mini pump with, DCZ coming out all the time.
We have time for one final question.
Hi, Lori. A couple of years ago, for completely different reasons, we did RNA-seq on brown fat following cold exposure. And in that paper, we reported that FGF21 and GDF15, those transcripts just go through the roof. And then the protein levels also go through the roof. And I was just wondering, this is certainly not an area that we focus on, but does that make sense as a feedback loop? Can you just comment on sort of...
Yeah, so this is something that we're very much interested, again, which is why time course matters here, is because a lot of the stimulations that have been looked at, like cold is obviously the most common one. Usually the cold exposures are allowed to happen for several hours at least. And when you do that, you do initiate a cascade of a lot of bradykines that are released, right? And then those will then act in turn on different organs, right? And so what we have done here, so first, the time frame that we see the stimulation is within like 2 minutes. So it's way before any transcriptional changes are going to have an effect. But what we are interested in looking at, and we're collaborating with someone to look at now, is to perform lipidomics in the context of, in the brown adipose tissue, in the context of the stimulation, to understand if you isolate the stimulation only to brown adipose tissue, what metabolites are changed versus when you have something like cold, where you have all of the organ crosstalk and, let's say, signals coming from fatty acids released from white adipose tissue lipolysis, for example. So now we can sort of start to tease out what's only happening via signaling via BAT directly versus how it's acting on other organs. And also by doing either lipidomics or even transcriptomics, in other organs, we may be able to get a sense of, so what if by just stimulating brown adipose tissue only, which other organs are you affecting directly? And so those are the sort of types of questions that we can now ask if we're just isolating that one part of the circuit.
But
Okay.
I guess what I'm wondering is, if you do have systemic FGF21 or GDF15 signaling, will that impact the ability of cold to induce brown fat thermogenesis?
So it's interesting because those are going to circle back. That what they really will do is they will impact, those two molecules will impact food intake specifically, and many other things, but that is also will drive is necessary for the adaptation to cold. And so I think, again, those types of hormonal sort of secondary molecules, I think, are really important for the longer term or even medium term, over hours, adaptations to cold. And that's why I think it's been so hard to tease things out, because in the manipulations that we're doing now, you're getting everything. You're getting direct effects on BAT, and then you're getting all the secondary effects due to the organ crosstalk. And so that's where we're hoping to sort of tease that out in the short term, 10-minute stimulation versus, a 3-hour stimulation or even a chronic stimulation.
Okay, thank you once again, Lori. Please.
Thank you.
Okay, now we move out to the first poster session. And before we conclude the morning, I also want to remind you to be back 11:15 for Professor Annette Ziegler's talk. And please also bring out glasses and plastic bottles. Thank you. See you 11:15.